Good morning, and thank you for joining us for today's webinar, Capsule Filling Optimization, a Guide to Performance, Troubleshooting, and Output Optimization. My name is Mikla Torsnader, and I'm the editor of Pharmaceutical Processing, one of Advantage Business Media's Manufacturing Group publications. I'll be serving as the moderator for today's event. Before I begin, I'd like to thank our sponsor, Capsugel. And I want to encourage everyone in attendance to take advantage of the interactive nature of this webinar platform by submitting questions at any time during the presentation. This can be done by typing your question into the corresponding box on your screen. We'll save all questions until the end of the presentation and attempt to answer them in the allotted time for Q&A. Please also note that there is a Q&A form at the bottom of your screen. Now to one of the reasons why you're all here. It's my pleasure to introduce Steve Rohde. With 29 years of experience in the biopharmaceutical industry, Steve is currently the Manager of Business Development for Capsugel Lonza Biopharmaceutical Group. During his time with Capsugel Lonza, he's worked with many top pharmaceutical and consumer healthcare companies in the development of pivotal capsule products such as Nexium, Effexor XR, Lyrica, OTC Prevacid, and Tafenor. In addition to his sales activities, Steve also had the opportunity to actively participate in various development projects and specialty teams with the company, including the development of DB caps, capsules specifically for use in double-blind clinical trials, VCaps plus HPMC capsules, and Connie Snap sprinkle capsules. I would also like to introduce Paul G. Davis. Paul is a Senior Technical Service Engineer for Capsugel, covering the West Coast, and has 28 years of experience in the pharmaceutical and herb manufacturing. He has worked as an operator supervisor and plant manager in the industry. He graduated from Utah Valley University with a degree in business. He's a certified Six Sigma Green Belt. Today, he works with hundreds of manufacturing companies on improving their processes, quality, efficiencies, and manufacturing packaging processes. He is married with his wife, and his wife has 5 children, and they have 3 grandchildren. Last but certainly not least, I would like to introduce John Davidson. John has worked in the industry for 15 years, holding a variety of leadership and engineering roles that have contributed to robust quality innovations at Capsugel. As part of his current role, he directs a team of quality engineers who design and support strategies for improving both biopharmaceutical and consumer health and nutrition manufacturing efficiencies. He is an engineering graduate of Georgia Institute of Technology and a Six Sigma Black Belt, and he serves on the Technical Advisory Board for Tablets and Capsules magazine, in which he has published several articles on improving manufacturing operations. So thank you all for joining us today, and I will turn it over to Steve.
Good morning, Meg. Thank you very much, and thank you everyone for joining us today and taking the time to be with us. In this section of the webinar, I would like to introduce various ways to think about the streamlining of the research and development activities through strategic use of capsules. We know a lot of research into new molecules begins in capsules and then moves away for various reasons into tablets or into perhaps other solid oral dosage forms. However, with a little bit of planning and some ingenuity, the use of capsules can actually be applied to an even wider array of molecules today than ever before. So let's start off the discussions with polymer choice, since this remains a question that we're always presented with. Most customers know the route of administration fairly on, and that knowledge is the first line to follow when choosing the best polymer for a situation. For instance, if we know that the polymer is through a pulmonary route, the best delivery, perhaps would be using an inhalation-based capsule. Based on that, we need to determine which capsule would be robust enough in order to be able to perform in a device that would pierce the capsule or cut it, and to allow the medication to be effectively evacuated out of the capsule. What we need to keep in mind, though, is the capsule choice and the polymer needs to be attuned to the formulation for successful product development, and then equally important, look at the tools for strategies to develop further. So if you know the route administration, if you know the properties of your API, then from advancing that into an oral route using a capsule, there are gelatin capsules, VCaps plus, enteric technologies, and even straight API into capsule options to consider. considering the actual choice of the polymers, as mentioned, the right capsule is extremely helpful in streamlining development. Basically, choosing the right capsule polymer means at the very beginning, I will not necessarily have to go back and repeat the studies if in the event the results I'm looking for are not achieved. In this chart, you can see a few very important capsules that are essential for early R&D studies, including a new enteric HPMC polymer that I'll spend some time on later in the presentation to discuss the details behind that. Included in the chart are several of the most important physiochemical factors corresponding to each of the capsules, as well as these factors and how they're important in choosing the right polymer. Consider that the release profile, for instance, is extremely important, whether it's for immediate release or if it's an acid-sensitive molecule, then perhaps protection from the pH of the stomach is critical. Today, more so than ever, in comparison to the past where gelatin was the only option for immediate release compounds, Capsugel has developed our VCaps Plus HPMC capsules, which offer several advantages over traditional gelatin capsules. While gelatin capsules have always been very robust and used for a multitude of purposes, we recognize and have always advised that hard gelatin capsules are kept within recommended storage conditions of 30 to 65% relative humidity, which helps to ensure that the moisture content stays in the optimal range of 12 to 16%. If conditions stray below, then we would see brittleness begin to come into play, which then can lead to manufacturing issues during the encapsulation process. In reverse, if the capsules are exposed to moisture and temperatures above the recommended conditions, then the capsules will soften, which in turn could lead to different types of manufacturing issues. As VCaps Plus capsules are made of HPMC and they don't rely on water content for pliability, the actual window of opportunity is greater than that as traditionally experienced with hard gelatin capsules. In fact, HPMC capsules can go as low as 2.5 to 3% moisture content before capsule brittleness is even introduced. And even at that low level, the potential for brittleness is still very, very low. So essentially, HPMC capsules contain anywhere between 2 to 9% water, which is on average 6% lower than that of hard gelatin capsules. In early studies in R&D, how hygroscopic or how moisture sensitive a molecule might happen to be is not always well defined. Therefore, if you're able to start with a capsule that has better moisture performance capabilities, this kind of levels the playing field and reduces the concern over brittleness and also the potential to have to use desiccants or other approaches to contain moisture or temperature variations with the product. When considering the use of HPMC capsules, however, it's important to realize that not all HPMC capsules are going to be equivalent when changing from gelatin. HPMC capsules can actually be made using two very different manufacturing processes. Traditionally, one which utilizes the gelling system, and then now the new process by which we use through thermogelation, which does not use any type of gelling agents whatsoever. As you can see by the charts, capsules that contain gelling systems can have a wide degree of variability based on the media used for their disintegration and dissolution evaluations. Another essential point to consider is VCaps Plus capsules do not contain any gelling systems. VCaps Plus, as mentioned, are made through a thermogelation process instead of the traditional HPMC capsule manufacturing process with gelling systems. Without gelling systems, the variability is considerably less, and therefore we see, looking at the in vitro dissolution protocols with our VCaps Plus capsules, excellent performance across the different media as pictured on the right of the slide in which we encapsulated caffeine into our VCAPS Plus capsules. However, it is known with most HPMC capsules that there is a slight lag time in the initial disintegration process in comparison to gelatin capsules. So to address these concerns, we actually conducted an in vivo study in which we compared VCAPS Plus capsules filled with extra strength Excedrin caplets and also ConiSnap gelatin capsules filled with extra strength gelatin with extra strength Excedrin. And the reason we chose the Excedrin extra strength, in fact, that it contains 3 different biomarkers, which represent 3 different BCS classifications. Thus, if there was going to be any potential issues as far as equivalency and release, having 3 different BCS products represented in one comparative study would give an excellent overview. So, if we actually look then at the different results from the study that we conducted, in the results, the acetaminophen marker is clearly shown that it is equivalent both in release in a gelatin capsule and also in our VCAPS Plus HPMC capsule. Here, gelatin is represented by the blue line, where our VCAPS Plus capsules are represented by the red line on the graph. And you can see that the performance is nearly identical with high confidence intervals. The same can also be said for the acetylsalicylic acid marker as well. Again, the gelatin is seen in blue, the VCAPS Plus HPMC is in red. When the values are examined in the table, the biggest difference was found in inter-subject variability, but otherwise the product performance was very close regardless of the polymer, as it also is shown with caffeine too. In each instance here, this BCS1 molecule, which has fast dissolution characteristics, there was roughly just a 5-minute lag time for the VCAPS Plus HPMC capsule, which showed no overall bearing in the in vitro performance between the 2 different polymers, and therefore had a very high confidence level between the 2 different capsule types. Thus, as you can see then, we've demonstrated HPMC capsules can really broaden what we would consider the working quality by design space for hard capsules, provided that you're utilizing HPMC capsules which are manufactured by thermogelation, which contain no gelling systems within the capsule. The lack of a gelling system greatly reduces the variability when seen with traditional HPMC capsules. And then by adding the in vivo data in comparison to gelatin, you see that VCAPS Plus really should be, for everyone, their initial first choice when starting formulation work. So in addition to utilization of HPMC capsules, VCAPS Plus, for immediate release compounds, there are, as we know, certainly compounds out there that require protection from the gastro environment and are highly sensitive to that pH range with the potential to degrade and to basically render the product non-efficacious. As a result, Capsugel in the last few years has spent a lot of time in the development of enteric capsules. And I'm happy to announce that, in fact, we have 2 separate enteric capsule options now available. We have our VCAPS enteric capsules, which we launched in November of 2016. And in addition, we have our intrinsic enteric drug delivery system, which we launched approximately 3 years ago. In both instances, these capsules provide enteric protection equivalent to that designated by USP. And I'll spend a little bit of time going into those performance characteristics that differentiate VCAPS enteric versus our VCAPS intrinsic drug delivery technology. However, before doing so, just a quick overview of how an intrinsically enteric capsule would mitigate risk, reduce costs, and save time in comparison to the traditional approach of either an enteric-coated capsule or an enteric-coated tablet. Certainly, as we know, enteric performance is required with products, and if so, it can take time in the development process to establish your approach to the enteric systems. That takes time. It could potentially mean additional time of 9 months through phase 1 and potentially 14 months through phase 3. And in the long term, these would be disruptive or extending time frames to getting your product onto the commercial marketplace. And if you look at the one summation at the bottom of the slide, albeit it could be contested, it is possible that by not having a product on the market, it could potentially be a loss of $8 million per day. So if you're able to save 1 month, 9 months, 14 months through your development process and commercialization, just by starting with an enteric capsule, this could very well get your product to market sooner and certainly improve the opportunity for long-term sales. The first enteric technology that we developed was our intrinsic enteric drug delivery technology platform. Essentially, this is a two-piece intrinsically hard capsule. The capsules are manufactured with pharmaceutical-grade cellulosic enteric materials. We currently produce those in a size 0 and a size number 3, and we do have the ability to extend this platform to other sizes and colors based on customers' requests. And these are utilizing systems that have been well known and well characterized in the pharmaceutical industry for over 20 years. As a result of being HPMC based, they are low as far as their water content, typically on average less than 4%. We do, however, as with all of our capsules, recommend keeping them under temperature and humidity conditions that are consistent with our gelatin capsules, but in fact, because they are HPMC, they can extend out to wider ranges of temperature and humidity exposure. So therefore, depending upon the zones that you're shipping your product into, this may be a suitable option of choice versus gelatin. We've also conducted shelf life and stability work in HTP bottles, and we're seeing no changes in the dimensions or enteric properties of the capsules. Fundamentals of this technology, what we do with our clients is we will enter into an agreement to work with you to formulate your product and put this into the capsules for you. So as a result, this is a development contract that we work by with clients as opposed to sending these capsules out empty for clients to evaluate in-house. One critical point to point out relative to our intrinsic is that the dissolution specifications for these capsules are exceptional. As everyone would like to have a 100% level of protection for their product encapsulated in our intrinsic, this example shows you that these capsules meet that. We've taken an extremely sensitive molecule, esomeprazole, encapsulated it into our intrinsic technology. This esomeprazole is the basic non-coated granules which are put into the intrinsic capsule and then put into the initial acid stage pH of 1.0, and then for 120 minutes moved over to the buffer stage at 6.8. As you can see, based on the performance, there is zero release of the esomeprazole during that 120-minute period. However, when introduced to the buffer stage at 6.8, you do see immediate release, and then with no less than 80% at time point of 150 minutes. Therefore, exceptional protection is provided. However, since this is an internal platform and clients would like to potentially initially evaluate their acid-sensitive molecules in-house, Capsugel has introduced just recently our VCAPS enteric capsules. This is also a two-piece capsule made of HPMC. In fact, it's a combination of both HPMC and HPMCS, and it is made using the same thermogelation process as we do for our VCAPS Plus capsules. Water content of these capsules is slightly higher than our intrinsic, on average 7%. However, we still do require and suggest that these capsules are maintained in the appropriate temperature and humidity conditions as all of our capsules. However, to keep in mind, these capsules we do provide on a sample basis. We will send them out to you based on your interest and execution of a CDA. So therefore, you have two options, both the intrinsic and the VCAPS enteric, for your acid-sensitive molecules. One thing of note is that HPMCAS is not allowed in food applications, so VCAPS enteric is specifically for pharmaceutical markets, not for health and nutrition applications. We have, however, established a DMF. We do have a C of A for this capsule, and we also have our technical reference file available as well, too. From a performance standpoint, you can see here we've taken Docalax tablets, which are commercially an enteric-coated tablet, and we have taken then the active ingredient, just a pure API, amorphous, and put it into our VCAPS enteric and run a side-by-side comparison, acid stage and buffer stage. And as you can see, the Docalax in our VCAPS enteric performed almost identical to the Docalax enteric-coated tablets themselves, therefore giving good protection for 120 minutes, and then when introduced to the buffer stage, you see the release taking place very, very quickly. Therefore, having a non-complex opportunity to avoid enteric coating and utilizing our enteric capsules, this can help save time and save money in your overall development process. Same exists for pharmaceutical product, Endocort. As you can see, we did a similar type of comparison with an encapsulated pure API versus the actual Endocort capsule with enteric granules inside, and you saw a similar profile being generated. Therefore, now that we're able to look at enteric solutions with capsules that themselves are enteric, you can determine whether or not VCAPS enteric is accessible for your needs, or whether the intrinsic would be better for highly sensitive molecules, both to heat and acid, such as peptides and perhaps molecules within the microbiome space. So now that we've made a selection relative to the empty capsule polymer, it's time to learn more from my colleague Paul, who will talk about the importance of training. Paul?
Thank you, Steve. Hello, everyone. I am going to share with you some real case studies where we found training and the optimization of encapsulation process to be beneficial. I'm going to cover in this presentation how training can improve your encapsulation process, how the training can help you with your encapsulation equipment, and through the optimization program, you can improve your encapsulation equipment so that you improve the quality, and the efficiencies of your production. So, starting with case one, Company A. Let me explain a little bit about this company. They are a large manufacturing company, had over 5 high-speed capsule filling machines. They were getting very poor results from these machines. They were running all their machines, but getting poor results. They were getting very poor output compared to what the machine should produce. Also, they were a company that had growth happening within the company, and they also needed to get more capacity. They could add more machines, but this requires more floor space, more capital investment, more labor, and so they wanted to look at other options. So they called Capsugel and asked us if we could come help. So we visited them and started evaluating their process. Using DMAIC, which is a Six Sigma tool or Six Sigma process, we look for opportunities to gain capacity for them and help them improve their encapsulation process. We found that they were running their encapsulation machines far below the OEM rated speeds. Some of the things we found is the operators chose just to run the machine slower, feeling that that would be better for the machine and for the output and quality of the capsule. We found that they had long changeover times. The reason for the long changeover time, it was the culture of the company. No one assumed that you could do it quicker. We found that they had other things eating into the changeover. Instead of focusing on getting their machines up and running the next product, they would get distracted with other things. We also found that they had splitting in their capsules, that they had quite a high defect level, and so that needed to be addressed as well. So with these findings, we put together a team, both with the company and Capsugel, to put some improvements in process. So we started a process improvement program. We felt training of the operators, maintenance, and we also had some of the R&D employees attend this encapsulation training. We felt that all those people being trained would help with that process. So the things that we trained them on were some of the things that Steve just covered for us. We helped them understand the importance of the size of the capsule and the characteristics of the polymer of the capsule and how they performed on the encapsulation equipment. Understanding that helps you to know how to set up and make certain adjustments on the machine. We also train them on the capsule filling machine, how to set up the machine, how to set it up correctly, how to do alignments on the machine once it was set up. We help them understand how to evaluate the parts and the tooling as they installed it on the machine to ensure that they were in good condition, not damaged or worn out. We help them understand the capabilities of the machine, how fast you can run the machine, the processes, or the way to understand how fast to run that machine. We also help them understand the importance of the vacuum and the air requirements to the machine, that if they are not set properly, they can cause issues in your encapsulation process. So, when trained in the correct setup and done correctly, it will help you with the quality of your production. When the machine is set up properly and you understand your formula and the type of capsule you're using, you can improve the quality, which helps with the purity and the safety of the product. Your scrap and defect levels are reduced. Also, your weight consistency, which is very important in the encapsulation process, that you maintain the proper dosage throughout the process, are maintained and stay in control. So, the results of our training of the operators, maintenance, and R&D, they were able to eliminate all their post-fill defects on all the products that they run. We were able to increase the speed of the machine to the spec levels for the equipment they were running. And if you remember from the first slide in this case, they were running at less than at 50% of capacity. So we automatically gained them 50% more capacity through this training. We're also able to reduce the time it took to change over the machine. So reducing that downtime also gave them more capacity with no additional costs. So with this training, with all this, not calculating the savings from not having to add more equipment, this resulted in a savings of $8.1 million annually that goes straight to the bottom line. This was money that they were spending and not going towards any profit to the company, just lost in the process. They were able to gain back for the company and get the capacity they needed to grow. Case number 2. We'll call them Company B. So where they're different from the first company, they were a medium-sized contract manufacturing company. They had a product that they had low profitability on. In fact, they were making no money on this product, and it was the largest product that they produced. They were under contract, so they didn't have the ability to go back and renegotiate that, or preferred not to without looking at other options. So they needed to cut costs on this product or become more profitable. So again, they called Capsugel to come in and help. So we went in, again, using the DMAIC process from Six Sigma. We evaluated the encapsulation of this product. What we found is that they were having a high level of bad joins or splits in the process. There's a picture there showing the example of one. The machines were running slow, 10% slower than the rated speed. And they were having to stop the machines often to readjust to maintain the dosage or the weight of the filled capsule in the process. And this was causing them to have poor output and efficiencies in their process. So through our diagnosis, we found that the equipment that they were using was old, not necessarily a bad thing, but it was older. We found that the parts and tooling on the machine was worn out, was damaged, had not been kept up, and that they were getting a high wear in the dosing tooling, or they had high wear in the dosing tooling because of the product, which was causing the frequent stops. Drilling down to some of the root causes, the older machine and parts, the worn tooling, this caused them difficulties in maintaining and controlling their dosing. We also, in the process, which hadn't been identified by them, was they were having poor loading of the capsules into the bushings on the machine. So it was an opportunity to make a capsule that was not utilized. And also they were having separation issues with the capsules on these machines, which again is an opportunity lost to fill a capsule and produce it. So just to understand that when a machine is not in proper repair, the tooling's not in good condition, it affects your yields. It causes you to have to reduce the speed of the machine to kind of overcome that. And you often are stopping the machine to make adjustments to try and compensate for the poor optimization of the filling machine. The effects of this are you have to have intervention with the operator, which is a risk to the safety of the operator. Again, you have poor capsule performance on the machines when they're not in good condition. You have poor control of your weight and the fill of the capsules. and you can have high defects. So we introduced to them an optimization program, which Capsugel has put together. Through this optimization program, we can ensure that when they're setting up the machine, the parts that are going back on the machine are as close to factory spec as possible. That they're in good condition, they're within those specs. We know that that's not going to cause an issue in the encapsulation process. We know that the inventory of parts are clean and don't have worn parts or damaged parts. If they have a crash on the machine and they go to get parts to replace something that might be damaged, we want to ensure that what they're putting back on the machine is at factory spec. It also ensures that the machine is set up properly, that in the setup there are no risks for defects or issues in the machine. It allows you to ensure that your vacuum and your air pressures are at the proper levels that are needed in the encapsulation process. It ensures that the machine is kept in good lubrication and prevents wear. And then the alignments of the parts, once they're on the machine, to ensure that they are aligned correctly, can all be done through this optimization program. So the results from this introduction of the optimization program. They were able to improve their yield by 4.6%. They had less machine stops because of the quality improvement, and also the ability to maintain the spec of the filled capsule. No longer were they having to stop and make adjustments in the process. They were able to speed up their machines to the full rated speed. Also, as I mentioned, some of the loading and separation issues, they were able to get that to 100% in the process, not missing those opportunities to fill capsules and produce at very high yields. So the result of this was they were able to find a savings of $950,000 annually on this product, which not only took them from a loss on this product, but to become profitable in running this product. So I hope you can see that in the encapsulation process, it is very important that the operators are trained in the processes and the correct setup of the encapsulation machines, and that your machines and equipment are kept in factory-like new condition through the optimizations of the machine and the optimization program. This will help with loss of profits, loss of money, and can help you gain capacity in your encapsulation process. So with that, I will now turn the time over to John Davidson. Thank you very much.
Thank you, Paul. And thank you, Steve, also for those thorough overviews. The title of this webinar is A Guide to Performance, Troubleshooting, and Output Optimization. I think Steve and Paul have laid some good groundwork on how important certain decisions early in the process are, as well as Paul addressed the impact of a standardized training program and really understanding your encapsulation process. My message is really going to be building on the foundation that they laid to this point and talk a little bit about quality risk management. Sometimes the best way to solve a problem is to avoid the conditions that led to that problem in the first place. We know in the pharmaceutical industry that quality risk management has become a key part of our total quality management systems, and within that, quality by design is something that we're talking about much more often nowadays. Borrowing from the ICH guideline Q8, pharmaceutical development, this really starts with that target product profile. And for our discussion today, I'm going to set the context of encapsulation. What was our target product profile look like if we want to optimize our encapsulation process? And if you read that guideline on pharmaceutical development, once you have your target product profile, then you must define what your critical quality attributes are. And that's what we'll do today in a very practical example. Then comes that word risk again, that risk assessment, linking those material attributes and process parameters, of course, back to the CQAs, defining the design space. Then it becomes a matter of control and continual improvement. So with respect to encapsulation, what is unique here is design space. That design space is defined by your equipment, your formulation. I think Paul laid out very well the three main factors in play in that consideration. Conceptually, your design space is a subset of what you know about your process. This is all predicated on having a scientific understanding of your process. One thing that I found through the years is that people will formulate very well for efficacy, a lot of focus there, but those formulators might not even know the type of filling equipment being used in the operations, or the selection of the filling equipment doesn't take into consideration some of those tendencies of the powder formulation that it is under consideration. So a scientific understanding of your process and what you're trying to achieve is critical. Then within that design space is what you want to define as your normal operating range, which should be smaller than your design space, hence the need for the control strategy. So as we talk about encapsulation and begin to narrow down this definition of design space, it's important to survey the options that are available to one when you're getting into encapsulation. There are small hand or manual fill operations that are available. There's semi-automatic machines that are available, and you can see in the chart the different scale of these pieces of equipment and their intended use. You have the automatic equipment, which is the largest scale, and that number of 300,000 gets tested each year as these machine manufacturers are developing new technologies. And then the opposite end of that scale is a more specialized application, perhaps where the accuracy of the dose is of critical importance, and you might be even microdosing in those instances. Now, each of these machines are built on different dosing technologies. Most of the semi-automatic machines have more of a gravity feed system where you're allowing gravity and the bulk density of the powder to fill that body cavity. Sometimes there's an additional auger feed that assists creating light compression. Then you have the tamping style dosing operation where you have a progressive compression of a plug over several stations, and then that plug is ejected into the body cavity. And then you have the dosator style where you're using lighter compression to build a plug, and then it's rotating and then dosing into the body cavity. So you can see that just through that description and the visualization here that there's different mechanisms in play that you must appreciate if we're going to talk about the design space of encapsulation. So if we go back to our quality target product profile and make that applicable for encapsulation, some things you must consider. Steve covered the capsule polymer requirements based on those formulary requirements. Perhaps it's bioavailability. Perhaps it's a sensitivity to moisture. Perhaps you're doing business in certain parts of the world where there's religious requirements that must be considered. That's one item that must come into play. There's the dosage strength in the regimen as well. Here I'm referring to your milligram strength and how many capsules per day do you want your patient to take. And then there's the intended dosing style of equipment. Are you gravity feeding, or using tamping system, or using a dosator? If we translate that now and bridge that to the CQAs, or the critical quality attributes, with respect to the capsule polymer, what's the delivery mechanism? What's the moisture content? What's my desired dissolution? Steve covered many of these aspects as he opened up the call today. I think he did a nice job. Then there's the dosage strength and regimen, which I alluded to is your milligram amount, the volume to deliver that milligram strength, very, very important, and then the appropriate capsule size for that targeted dosage regimen, or your capsules per dose. And we'll spend just a minute explaining this one a little bit further in this section. Then with respect to the dosing, that maps over into the physical characteristics of formulation. So I mentioned before that we're formulating for efficacy first and foremost, and that's the absolutely right approach. But there must be a consideration for those physical characteristics in order to optimize our outcome, and we'll talk a little bit about a case study here as well. But some of the critical items to consider here are the lubricity, the flow, the compressibility or cohesion of that powder if you're going to make a plug. So stopping first for a moment on choosing the right capsule size, this might be one of the most often asked questions, and also one of the things that I always encourage consideration of early in the development process, because the impact is very great if you choose the wrong capsule size. There is information available. There's a Capsugel technical reference file that we can provide each of you. The appropriate capsule size or the volume that it will maintain is in a chart. The delivered powder density, and the reason I say delivered, the powder density as it will be dosed should be measured. And then you can make a volumetric calculation using this chart. And for this example, that bulk density of that delivered is that 0.81 grams per cc. For a target fill weight then of 380 milligrams per capsule, the chart shows that size 1 capsule would be the most appropriate fit in this case. The target fill of 380 is about 95% of that 400 milligram capacity, which is an ideal choice. The mistake that can be made here is having a plug that extends far outside of the body portion, and that can lead to defects in encapsulation and even further downstream impacts if not considered early. So mapping then those formulation physical characteristics, just a little bit of more information here. Lubricity is needed so that you can ensure a clean ejection of that plug from each of the dosing styles into that body cavity. The flow of the product is important for the weight uniformity. As the powder is dosed, it must fill the voids. The flow of the powder and the time that it has to flow are both important. How compressible the powder is another quantification that one can make, and is very important to ensure cohesion so that you don't have a plug that breaks apart during ejection and then spills over into your segments or overflows the body because you don't have a clean ejecting plug. All are very important. And there are ways to measure these, and I'll show that just in one second, but mapping that back over to your dosing style considerations. If you already have equipment and you are wanting to formulate to that equipment for a gravity feed system, it doesn't rely on high density or compression. It's a pure gravity feed, so the uniformity of the powder will be important, but you rely mainly on gravity here to do its work. From a tamping standpoint, you want to have good material flow. You want a medium-high density and a medium-high compressibility so that you can build a cohesive slug and eject it from the machine. With dosator, you're using less compression, so cohesion attributes are one of the most important. So again, the plug doesn't break apart upon ejection. There are tools commercially available to quantify these key quality attributes. A Scott volumeter will help measure the apparent density of a variety of free-flowing powders. The tap bolt density analyzer helps measure that bolt density under the influence of an oscillating gravitational force with some light tamping. And then the slug tester is a great tool to have as well, as you can determine the actual dosing disc height and capsule size based on the compression applied through this tester on the material that you're going to be actually dosing. Indispensable tools to have a part of your operation. Again, we typically formulate very well for efficacy. The addition of these tools mean now that we're also formulating to optimize our outcomes on the encapsulation equipment itself. And just to illustrate that point, I once worked with a pharmaceutical company, and they were experiencing complaints for a bitter taste. Very curious for them to get this complaint because they were using capsules, and capsules are great at taste masking. So when I got involved with this particular project, I also found out that they had to slow down their filling to maintain their weights. They had high yield losses, a lot of powder loss on the table, and a fair amount of downtime involved in having to clean up during the shift. The culprit was a defect that actually Paul previewed with you. Now, this is not the actual defect, using a capsule that I can show you so we can protect the innocent. The severity of this bad joining, as we refer to it, was such that even after dedusting, immediately after encapsulation, the bad joining had created a track for the powder to escape even over time and even after the packaging. The reason that they had the bad joining defect is during the closing of the body and the cap portion on the equipment, that powder that was all on that machine was interfering with the closing and causing that fold over. That bitter taste the consumers were experiencing was from that residual powder leaking and getting on the machine. So the powder cohesion characteristics were not adequate to form a plug in this case, and so it was a very, very loose plug that was made. To achieve the desired fill weight, the product was filled beyond its capacity. Here's a great example of the capsule size not being considered for the actual bulk density of the dosed powder, and so they were overfilling. That poorly formed slug, that undersized capsule, resulted in that powder loss that caused that bad joining, which allowed the powder to leak, which impacted the patient with the bitter taste. In this case, we're very, very fortunate in that the compaction or compression during tamping could be improved without impacting the dissolution of the product. So in this particular case, we were able to assist and help through formation of a better plug, thereby reducing drastically the powder losses. But they didn't understand the importance of the cohesion in that plug, therefore the machine was not set up adequately, and therefore they had the downstream issues. Now, beyond the customer complaints, there was a material, a financial loss associated as well. And once that plug was resolved, there was a significant decline in their consumer complaints, a 10% improvement in yield, less downtime, and they were also able to run their machine closer to the rated speeds. The estimated financial savings by resolving this problem through that machine adjustment was in excess of $750,000, so there was a lot of money in play as well. But I hope that case study illustrates the power of understanding your process and the opportunity to avoid those types of downstream impacts of not formulating for the equipment that you're using or not adequately setting the equipment up for what you need to achieve. Now, quite often I'm asked, well, how do I get started in this? I've got several formulations I need to evaluate. I would encourage you to get some of those essential tools that I spoke about earlier. Use those tools to properly assess the physical characteristics of your formulation. Of course, make sure you've got the right capsule size, and go ahead and develop a collaborative relationship with the operations team. Formulators should be talking to operations. Quality should be a part of that triad as well. Develop pilot programs so that you can do small batch and scale up. Also do periodic reviews. The formulator can be as much responsible for the yield on the equipment as the operations group when it comes to applying these quality by design techniques. which of course means begin with the end in mind and design success into your formulation. So that's one way you can mitigate risk using quality by design techniques for your encapsulation department. Another way that you can mitigate risk is through vendor partnerships. Quality agreements are always a great place to start, clarifying expectations, making sure contacts are clear. Qualified vendor programs are one step above. Great thing about qualified vendor programs is typically there's a risk assessment involved, and it can lead to reduced testing for your low-risk components, excipients, enabling your testing resources to focus on higher-risk raw materials. So I strongly encourage qualified vendor programs because there's a financial savings involved there as well. Look for risk-mitigating technologies. Capsugel recognized the risk adversity in the market with respect to physical defects, respect to visual defects, and developed what we now call the ConnieSnap Sigma series, which moves well beyond acceptable quality levels, which our industry has traditionally run on, and has moved to Sigma levels, which measures in parts per million, significantly reduce risk that you can plug into your quality risk management program. Particulate matter is another item that we're discussing more and more. Now there are packaging options to reduce the amount of corrugate fiber you're introducing into your operation, such as fiber-free packaging as depicted in the picture there. And lastly, risk also takes the form of supply. And I encourage you when you look at security of supply and you look at the supply chain, it's not just about getting product on time and in full. It's also available capacity. Most people want their company to grow. Can your vendors scale with you? How fast can they activate capacity? It's integrity of supply. How well are your vendors or your suppliers managing their suppliers and the raw materials that go into their products and components? It's business continuity. What type of footprint do they have? Is there a good business continuity plan not only for you but for them? And of course, responsiveness. Is the product getting there when you need it? Do you have the lead times that you need? Do you have business enabling supply chain functions that make that possible? So that is a broad view of ways that you can manage risk. Hopefully a few practical examples that you can take away and use in your operation. I think we're nearing the end of our time together here, so at this moment I'll hand it back to Meg.
Thanks again to John, Paul, and Stephen for your time today, as well as our sponsor, Capsugel. So if you'd like to take another look at the webinar or share with a colleague, it will make sure to do so starting tomorrow. But while we still have a few minutes, let's start with a Q&A. Our first question will be directed to Steve. Are the HPMC capsules form cross-linking on stability as in the gelatin capsules case?
Thank you, Meg. No, they do not. Unlike gelatin capsules, which are susceptible to cross-linking based on temperature and API exposure and those types of things, principally because gelatin capsules contain amino acids and proteins, HPMC capsules are not susceptible to cross-linking.
Perfect. Thank you for your insight. Our next question will be directed to Paul. Does Capsugel see non-OEM parts on a capsule filler causing performance issues?
So to answer that, what is critical here is that the tooling that is purchased is to the spec of the OEM. So we make our capsules to meet and understand spec that we collaborate with the manufacturers of the machine so that the two together will work properly. If you are buying tooling that is not maintaining that spec bore to bore, part to part, or they've already are worn out or not as tight as they need to be, that you're actually lowering the life of the tooling or opening yourself to risk. So I'll just answer that by saying know what you're buying, know the quality of it, but you can't go wrong with buying from the OEM from the original manufacturer's tooling.
And our next question will be directed to John. Are the potential savings implementing the optimization program relayed to the customer as an investment versus savings report?
Good question. So the optimization program is designed to prevent issues from occurring in your process. So quite often we work with customers to help quantify the potential savings or the cost avoidance using that calculator that we showed a few times. We built that calculator based on some industry standards, and it can be completely customized to your very specific need. And many of our customers use that as a return on investment calculator also in the case that a bad segment needs to be replaced or repaired. So we've used that as a savings. We've used that as a return on investment calculator. But if you have a more specific question or desire to use that approach, please reach out to us. We'd be happy to help.
Okay, perfect. And this will be our last question of today. This is for Steve. Can you describe the differences between the common HPMC gelling process versus Capsugel's thermogelling?
Sure, absolutely. So the traditional HPMC capsule manufacturing process, which utilizes gelling agents and gelling promoters, you're essentially adding those into the dipping process when the capsules are being formed so that the HPMC will produce a hard physical shell. As mentioned in the presentation, there's a lot of variability associated with the addition of gelling agents and gelling promoters. So what we've actually done is devised a proprietary process based on thermogelation, where we're controlling the temperature of the HPMC and the dipping process so that instead of having to use the gelling agents and gelling promoters, the capsules will physically form just as they form in the case of gelatin capsules, which also don't contain any gelling agents or gelling promoters. So the traditional process when HPMC capsules were first introduced absolutely required those two components in addition to the HPMC, but because of our proprietary process, we dip and manufacture our VCaps Plus without any gelling agents or gelling promoters, just in a manner similar to gelatin.
Great, thank you for your insight. So thanks again to John, Paul, and Steve for your time today, and our sponsor Capsugel, now a Lonza company. If you'd like to take another look at the webinar or share with a colleague, it will be available for on-demand viewing at pharmpro.com starting tomorrow. And that concludes today's presentation. Thanks to everyone for attending, and have a great day.